Hi, my name is Spencer Hay. I'll be talking today about visualizing the clinical research landscape and why it matters for efficiency, transparency, and ethics. Before diving in, I'll start with a few details about myself and my history. I spent over a decade in academia doing my graduate work in philosophy of science before transitioning to clinical research ethics and science policy. I worked first at McGill University up in Canada, and then moved to Harvard Medical School in Boston, which is where I live now. About a year ago, I started transitioning away from academia to found my company, Prism Analytic Technologies. So I should acknowledge here at the outset that I do have a conflict of interest related to what I'm going to talk about today. In fact, I started my company to try and overcome many of the challenges I'm going to talk about. While I do hope that by the end of this talk, you will think that Prism is doing something pretty cool and important, that is not the primary goal. The primary goal is rather to convince you that efficiency, transparency, and ethics in clinical research are mutually reinforcing. So when we talk about these three concepts, particularly in the context of drug development, you will often hear people say that ethics and efficiency are in conflict because, for example, we can make clinical trials faster and less expensive if we don't have to collect informed consent from all the participants. Or sometimes people will argue that transparency and efficiency can be in conflict because it is expensive and time-consuming to upload and make all the data from a clinical trial publicly available. Some people will sometimes argue that if patients were really sufficiently informed about the risks, burdens, and realities of clinical research, they might not even want to participate in trials, and so the whole research system would grind to a halt, in which case transparency and ethics are in conflict. Now, there may be some truth in all of these views in the sense that they stem from some of the existing realities and challenges for the clinical research enterprise. But today, I want to paint for you a different picture, a picture of how I think clinical research should be, a picture where efficiency, transparency, and ethics really do go hand in hand, and where the entire field of clinical research can proceed in a way that can better benefit researchers and investors and reduce unnecessary harms to human subjects participating in studies. So in my talk today, I'm going to show you three pictures of the research enterprise, and I will argue that each of these pictures helps us to see how efficiency, transparency, and ethics work together. So here we go. Picture one, a drug development trajectory. So a little bit of setup. Around 2008-2009, the Tuberculosis Trials Consortium, or the TBTC, was trying to decide if they should launch a phase 3 trial involving a new drug called moxifloxacin. So phase 3 trials are typically the most expensive trials in a development process. So they cost tens or even hundreds of millions of dollars. They can take many years to complete, and they usually involve hundreds or thousands of patients. So when most people think about clinical trials, it is typically these big phase 3 randomized trials that they have in mind. A phase 3 trial is also considered the gold standard to show the new drug is effective, and regulators will typically require at least one positive phase 3 trial before they're going to approve a new medication. But because these trials are so expensive, it is often a fraught decision whether to initiate one. Indeed, the jump to phase 3 in a drug development trajectory is known as the go-no-go decision. Are we going to place a huge bet on this drug, or are we going to put our resources elsewhere? So when the TBTC was faced with this go-no-go decision for moxifloxacin, they had a lot to think about. There were already 5 phase 2 trials, and these are smaller studies that are useful for generating signals of a drug's efficacy, but aren't considered decisive tests of its clinical utility. So for moxifloxacin, the 2 largest phase 2 trials showed that moxifloxacin wasn't beneficial for treating tuberculosis, and yet 3 smaller trials suggested that it was. So because I had the good fortune to be working with one of the TBTC statisticians at the time, I was sitting in the room at the scientific meeting of the TBTC's annual conference, and I listened to the scientists debate with each other about what to do next. Was a phase 3 trial a good investment or not? So now let's look at the picture. So this is from a publication of mine that came out a few years after that TBTC meeting. And in this paper, I presented a graphical method for describing this kind of decision point. And I call this an AERO graph, so AERO, which stands for Accumulating Evidence and Research Organization. And in essence, an AERO graph is a categorical scatter plot that depicts the evolution of evidence over time. Each node here corresponds to a study of moxifloxacin for treating tuberculosis, and it's arranged by the study's date along the x-axis and its phase along the y-axis. Then each node is color-coded to reflect the direction of its outcome. Positive studies, which show that moxifloxacin is beneficial for treating TB, are colored green. Negative studies are colored red. Studies with equivocal or ambiguous outcomes are colored yellow. Then in this figure, because I worked closely with some of the researchers, I've also added arrows between the nodes to reflect the intellectual lineage. So the idea is to capture which results inspired which next studies. So in this way, we can tell the story of the evidence for any given decision point. Where are we today? How did we get here? What are we thinking about doing next? So if we look here between 2006 and 2009, we can see the 5 phase 2 trials and their discordant results. And I've also added these blue-lined nodes with capital letters to reflect the possible next steps, as indeed these were the options that the researchers were considering. Some argued that discordance between animal and human trials suggested that option A was the right next step, to work toward improving our understanding of the animal models. Some argued that toxicity and drug interaction signals needed a closer look, and so option B, another phase 1 trial, was the right choice. Some argued that another phase 2 trial could perhaps tip the balance and clarify whether the drug is really ready for phase 3, so that's option C. And some argued that moxifloxacin looked promising enough already and had not been ruled out by phase 2, and therefore proceeding to phase 3 was really the right way to go, and so that's option D. Now, I argued in this paper that this method was not only useful for eliminating the go-no-go decision for the TBTC, but that it was also completely generalizable. Any drug development trajectory can be represented in this way, and the picture can then help to clarify scientific reasoning and broker points of agreement and disagreement. But of course, the scientists at the meeting back in 2009 did not have this picture. Instead, they essentially pounded their fists on the table and talked past one another, each offering recommendations for next steps, but without a shared concrete reference point for how they were interpreting the evidence and why that evidence justified their preferred path forward. And as it turned out, just as a denouement to the story, the community did go ahead and run the phase 3 trial, but that trial ended up having to be terminated early because of excessive harm to the patients in the moxifloxacin arm. But now let me come back to my thesis and talk about how I think this picture helps us to see the ways in which efficiency, transparency, and ethics can work together. Imagine for a moment that the TBTC researchers had put up the picture I just showed you at the start of their meeting. I contend that this would have made their strategic deliberations far more efficient, because it focuses their discussion around a shared map of the evidence, allowing them to put their fingers, so to speak, on particular pieces of evidence and points of disagreement. It also makes the justification for what to do next more explicit and transparent. This helps them to study their own process, since they can then look back over time and examine how they reason from evidence to action. And if things don't pan out how they hoped, like the phase 3 trial that terminated for harm, now they're in a stronger position to look back and learn and improve their decision making for next time. Furthermore, once a decision is made, this picture and reasoning can then be shared with others, like patients and ethics committees, to clarify why the particular trial is safe and valuable and exactly the right study to be doing given everything else that we know. So in this way, the picture helps to promote transparency, which promotes ethics, which promotes efficiency in drug development. Yet there are some limitations that should be acknowledged here as well. Getting this picture prepared takes time. Now, I did this work back when I was a graduate student, and it took me months to gather all the data, talk to the researchers, and painstakingly write up the code in LaTeX to produce this image. Having the arrows between studies also requires some insider knowledge. And this can be hard to come by, or sometimes even impossible to come by, if the researchers who do the work don't want to talk about it. Scientific evidence is also continuously evolving. Since the picture I just showed is static and was only made public in the context of an academic publication, it can only reflect a snapshot of where things stood at the time of the analysis. And this may be out of date by the time the picture appears in print. But perhaps most importantly, knowing what is the right next step to take should probably also appeal to evidence from other development trajectories. So for example, what is the background rate of success for all TB drugs? How many times in the past has a successful TB drug showed inconsistent efficacy in phase 2? Properly justifying a decision in any particular instance surely requires this kind of contextual understanding, which requires taking a broader look at the research enterprise. So now let's take a broader look at the research enterprise and consider a cluster of drug development trajectories, or what I call here a drug development portfolio. Now here's the setup for the second case. Cholesterol ester transfer protein inhibitors, or CETPs, are a class of drugs that entered clinical trials around 2001 with a seemingly solid biological hypothesis and great clinical promise. These drugs raise a patient's high-density lipoprotein, or HDL, the so-called good cholesterol. The hope is that raising HDL with a CETP inhibitor would lead to a reduction in cardiovascular risk, similar to how lowering low-density lipoprotein, or LDL, or the bad cholesterol, with a statin is associated with a reduction in cardiovascular risk. Unfortunately, one after the other, the CETP inhibitors failed to show benefit in clinical trials. One member of this class, torcetrapib, even increased the risk of serious cardiovascular events, and this came as a pretty big shock to the pharma community. However, in 2017, Merck's phase 3 trial of anacetrapib, which was the fourth CETP inhibitor to enter phase 3, did indeed find a small but statistically significant benefit. Yet shortly after that trial's result was published, Merck announced that they were abandoning the drug anyway. So let's sum this up. This is almost 2 decades of clinical research, billions of dollars spent on development, tens of thousands of patients enrolled, and we seemingly come out the other side of this process with nothing. So what's going on here? Now here's the picture of this drug development portfolio. This shows 5 different CETP inhibitors arranged alphabetically by drug name and phase along the y-axis, and by study publication date along the x-axis. Just as in the moxifloxacin picture, the color of the nodes here represents the study's outcome. Positive is green, negative is red, equivocal is yellow, and gray nodes are studies whose results are unknown. The size of the nodes is now also doing some work, as it corresponds to the number of patients enrolled. So these big nodes are the large randomized phase 3 trials, each of which has more than 10,000 patients. The figure is also now interactive, so I can zoom and hover and click to drill down and get more information about each of these trials. So there's a ton to unpack here, but for this talk, I just want to highlight a few key things. So first, this picture shows us that almost every phase 1 and phase 2 trial with these drugs was positive, and yet 3 out of the 4 phase 3 trials were negative. This repeated pattern of positive early phase trials followed by negative pivotal trials suggests that the early phases may not be giving us good signals about what is likely to benefit patients. Indeed, the vast majority of these early trials were using changes in lipid levels as their primary outcomes, but it should have become increasingly clear over time that a drug's ability to modify lipid levels does not necessarily translate into an ability to reduce cardiovascular risks, and that's the primary outcome in these phase 3 trials. Second, because this portfolio is all based on a shared hypothesis about how these drugs are effective, the hypotheses tested in these trials are actually correlated, which means that we must interpret the results of each individual trial in light of every other trial in the portfolio. So even though each of these drug development programs is run by a different company, the results of the 3 prior negative phase 3 trials necessarily influences how we should interpret Merck's big green successful trial, for example. In essence, because the hypotheses across these trials are correlated, we're supposed to shrink the estimated effect size towards a common mean effect. And while I can't say for sure, I think this is actually why Merck abandoned the drug despite the positive phase 3 trial. I think the FDA looked at the whole portfolio and told them they would have to do another trial to confirm the result, and Merck decided that it just wasn't worth it. And this brings me to the idea of the point of diminishing returns. From a pure science perspective, there's nothing wrong with testing the same hypothesis or family of hypotheses over and over again. Indeed, this kind of scientific perseverance is sometimes critical to achieving important insights and breakthroughs. However, clinical research is not pure science. These are incredibly expensive experiments with huge opportunity costs. These are also human beings that we're experimenting upon, and we are ethically required to redeem the risks and burdens we place on the human subjects with commensurate gains in scientific and social value. So for me, this is an incredibly instructive case and picture, because it helps us to put a fine point on the question of when we, as a scientific research community, already know enough about a particular drug or class of drugs that we ought to turn our attention elsewhere. Okay, so come back to the refrain. What does this picture of a drug development portfolio teach us about efficiency, transparency, and ethics? So in terms of efficiency, I would argue that pictures like this can help us to spot emerging patterns or results across a portfolio that we can then use to design more valuable studies or prevent unnecessary studies. In terms of transparency, I would argue that pictures like this should be a standard part of how we all communicate with each other about clinical trials. Just like how I think the TBTC researchers would have benefited from a shared picture of the moxifloxacin trajectory, the companies, regulators, ethics committees, and patients in the CETP case, I think would all have benefited from being able to reference this picture as they made decisions about whether to initiate a new trial, to approve a particular study protocol, or sign up to be a trial participant. And this feeds directly into ethics. As I mentioned a moment ago, trials are ethically justified only if their risks, costs, and burdens are outweighed by future benefits to patients and society. Each study in the CETP inhibitor portfolio needs to satisfy this requirement. So if it's the case that we do go past the point of diminishing returns, then this becomes an ethical problem. So again, ethics demands efficiency, which demands transparency. But let me hit on some limitations here as well. So this interactive figure is coded in Python, and while it took me far less time to create than the moxifloxacin trajectory, it was still something of a painstaking multi-week process. There are also some interesting conceptual issues here, I think, like what are the correct bounds around a drug development portfolio? How do we decide which trajectories should be grouped together and which hypotheses are correlated? In some cases, like with the CETP inhibitors, it may be sort of obvious, but in other cases, like, say, cell and gene therapies, for example, it may not always be so obvious how to classify or group different, albeit related interventions. Past patterns of success and failure also do not necessarily predict future results. So good judgment is still required to know how to draw reasonable inferences from the patterns that are made evident by these kinds of visualizations. Thus far, I've also been showing trials as existing at the time of publication, but of course, trials are not frozen points in time. They are actually taking place over time. However, this limitation is truly just an opportunity, I think, to show another picture of where I see the future of this kind of analysis going. So here's an animation of this same data, where we can see the trials appearing and growing and resolving over time. And we can even start to scrub the timeline to really start to put a fine point on the question about diminishing returns. Was there an earlier time when we really already knew enough to predict what was going to happen next? Now for me, I think 2012 is probably that time when, based on the accumulating evidence, we could have looked at everything that we knew and predicted that this class was not going to pan out. Now that's hard to say for sure, of course, and hindsight is 2020, but I contend that efficiency, transparency, and ethics would all be promoted by having a system in place that would allow us to explore the clinical trial data in this way as a matter of course. But then, just like our understanding of a drug trajectory is likely incomplete without its surrounding portfolio, it stands to reason that our understanding of a drug portfolio is incomplete without an understanding of the even broader context. And that brings us to picture 3, looking at these issues at the enterprise scale. So this last analysis uses essentially the same Python code that I used for the CETP inhibitor portfolio, except here I've pulled the data on all the clinical trials from 10 large pharmaceutical companies and graphed it on one giant landscape. The trial start date is the x-axis, and the y-axis is the condition or patient population of interest. And the colors of the nodes in this case indicate the company rather than the outcome. The size of the node is still the number of patients enrolled, and the shapes here indicate the trial status. So circles are completed studies, circles with an X were terminated, triangles are the active and recruiting trials, and squares are everything else, so that's studies that were withdrawn or studies that aren't yet recruiting. Now looking across the entire enterprise in this way, what can we see? Well, we can see that different disease areas look really different. So here are some infectious diseases and malaria, where we see just a couple of companies active, but they are conducting these enormous trials. And this trial size makes sense given that many of these are testing vaccines and are public health trials, and this really requires large numbers of participants. If we look at some neurological conditions by contrast, we can see that things are really different. So these trials are smaller, but we also see more companies active in this space. Alzheimer's disease is clearly denser than Parkinson's, and in migraines we see this sort of intriguing drop-off in activity around 2011. Or in meningitis, really only one of these 10 companies has been active, but they've been pretty consistently running trials for the past 15 years or so. If we look at cardiovascular diseases, we can see there are many companies in this space, all conducting very large trials, just like we saw when we were zoomed in on that one drug portfolio. And we can also see many terminated trials here. So terminations are not necessarily a bad thing because trials can be terminated early when a drug is much more effective than anticipated. But generally speaking, trials are not designed to terminate. So a lot of terminations suggest that the research community may be consistently off in their judgments about what interventions to test and how best to test them. And here we can see cancer, which again has its own unique character. Many small trials, many different conditions, which makes sense given that cancer is not one disease, and as the number of cancer indications proliferates, the eligible patient populations to study are necessarily smaller. We can also see a lot of triangles here, so a lot of consistent activity in this space. And particularly for breast cancer, colorectal, lung, and prostate cancer, we see pretty solid trial activity for the past 20 years. Now this may be a good thing because these cancers are all serious and we still need better treatments, but we might also wonder if it would be better to see activity become more widely distributed over time. So for example, maybe what we would want to see is periods of intense research followed by gaps as the companies succeed in developing cures in one area and then shift their focus to new areas with greater needs. Okay, so I think many of the advantages for efficiency, transparency, and ethics here are the same as those I've already mentioned. This is really just looking at the same kind of data again, but really at a higher scale, by zooming out another level or two so that we get this greater appreciation of the context, and that gives us more information and insight to draw on when making decisions about what we want to do next. So what questions have already been saturated with research? Where are the gaps and opportunities to make the biggest difference? But let me just add a few more ideas to the mix. First, I think it is vital to appreciate that this picture of the enterprise, which shows an incredible volume of activity in clinical research, also contains within it the life's work of thousands of scientists that was actually only made possible because of the millions of patients that essentially donated their bodies to this effort. And in this way, this story that's told by this picture truly involves all of us. Literally everyone on the planet is affected in some way by what is happening here. Some people are affected more directly than others, but everyone is or knows someone who has been affected by at least one of these diseases. And everyone knows someone whose life was transformed because of a breakthrough in one of these trials. And to me, that is part of what is profound about this picture. This gives us a new perspective on what I think is a pretty incredible human achievement. This is a massive investment of social resources, all devoted to the science of making our lives healthier and better, constantly evolving and growing and changing shape as we learn more and more about our biology and our well-being. So let's start putting the pieces together. Each picture that I've shown you today represents one layer of a powerful scientific platform. Imagine being able to survey the whole clinical research landscape, look for gaps, redundancy, opportunities, and so on, then select particular questions or research portfolios, examine patterns of development and drill down on individual research trajectories, then zoom all the way in to examine individual study results, and zoom all the way back out. All right at your fingertips, all with the click of a button. And the technology is really already there. That giant picture of the research enterprise that I showed you, that took me only a few hours to construct. All that's needed to build this system is really the vision and the dedication. And this system, it seems to me, opens up a whole new set of scientific questions that we're not even thinking about asking until we start to see and interact with the information in this way. So what does a well-ordered science look like? How should we be shaping the future of clinical research in healthcare? How can we incentivize the scientific community to start making more data-driven, transparent, and ethical decisions? To me, these are some of the most challenging and exciting questions, but without the understanding provided by these visual perspectives of the research enterprise, these questions are all sort of metaphorical in character. But once we have this understanding and perspective, now we can actually begin to answer these questions in a really rigorous way. So that's what my company is doing. Now remember, I told you at the beginning that I had a conflict of interest here, but hopefully I've also convinced you that this is important and exciting work. At Prism, we are building this system to refine and scale up the research and data visualization tools that I've just shown you. And our goal is indeed to be a part of making the research enterprise more efficient, more transparent, and more ethical. Here are some references to the peer-reviewed literature behind everything that I've just shared. And I want to thank you for your time and attention. And if this is exciting to you, I'd love to talk more. You can get in touch with me at spencer@prism.bio. Thank you.